19. And this is an example of how your immune system uses selection, natural selection, to improve the immune response. And of course, your immune system is the product of many millions of years of evolution. Antibodies are shaped like Ys. This is a antibody, and the part where it binds the thing that you're allergic to that's called an antigen, and that's at the tips of the Ys. Those are the binding chains. And here's an antigen molecule bound to that antibody. And one of the absolutely amazing things about the acquired immune system, which is your antibody, where your antibodies come from, is that DNA can rearrange itself right at this bit. The DNA that codes for the protein that makes this shape right at this bit where it binds the antigen. It can rearrange itself in random ways and make shapes that are compatible with antigens that you necessarily haven't even seen before. For example, like COVID-19, which didn't exist on the planet last year. And it can adapt for those. And in some studies in the 60s, where they're looking at antibody response, this is in guinea pigs. So a literal lab guinea pig. They looked at the difference between the primary response and the secondary response in terms of primary production of antibodies after the guinea pig was immunized with an antigen. Exposed to an antigen, usually injected in the blood. And I'll have you take a notice of the y-axis here, where you have the concentration of the antibody, and it goes from 0 to 60, and it's times 1,000. And then that response and how it goes after days. Initially, there's pretty rapid response, and then you get tapering off of response after 10 or so days. If the same individual guinea pig is immunized with the same antigen later on, say a month or two later, you have a much stronger response. So notice this scale goes from 0.1 to 60, and this scale here goes from 10 to 2,000. So even though this physically is lower than this curve, it's a much, much higher response in the secondary response. So the first time you're exposed to an antigen, you don't make as many antibodies typically as the second time. Now I'll say this really differs depending on the antigen. This is part of the question that is yet to be answered by our current pandemic, is when people have a primary response after they're first exposed to the antibody, the COVID-19 virus, are they making enough antibodies of the right kind that can inactivate the virus? So these antibody tests can detect antibodies in people who have been exposed to the virus, but are they in high enough concentration, and are they the right kind of antibodies, so-called neutralizing antibodies that would block further infection? Okay, if we look at these initial response, the primary response is relatively weak compared to a secondary response, 100 times stronger response the second time these guinea pigs were immunized with the antigen. Okay, then another question was, well, that's a cute little experiment, but that typically is not how organisms encounter antigens. It's not neatly one at a time in the environment. Typically, they happen more than you get more than one antigen at the same time. So another group of scientists working with rats this time looked at the primary response and secondary response, and in the secondary response they gave two different antigens, but then they only measured the response to the original antigen. So the idea was, okay, I'm going to immunize these rats with something and get a primary response, and the primary response was typical, that it took about 10 days after injection before they had immunity, and it was not a fantastic amount of antibody, but some antibody. And then the secondary response, they gave the rats more than one antigen, including the original antigen and then a second antigen, and then they only measured antibodies against the original antigen. Wanted to know if it got screwed up or faked out by the second antigen, and they didn't. They had a typical secondary response. So after that, a lot of the responses are independent. The primary and secondary responses are independent. After that, a lot of cell biology was done, figure out what's going on. So your cells in your immune system that make antibodies are called B cells. B because they are found in bone marrow. And these B cells will make antibodies in response to binding an antigen through a process called clonal selection. And then those antibodies, once they're stimulated by the antigen in clonal selection, are those B cells, once they're stimulated by antigen through clonal selection, will go through mitosis, and the antibodies will secrete or export these proteins outside the cell, and then these antibodies can float in the blood. Antibodies can bind to infectious agents and neutralize them, prevent them from getting into cells, like if they bound to the spike protein on the outside of the coronavirus, and or if they bind to things, they can activate white blood cells, which will come eat the thing where an antibody has tagged it. But B cells can also make memory B cells, and these are involved in the secondary response. And those memory B cells are hanging around, and if they're stimulated by the same antigen later on down the road, they have a much bigger response in terms of making many, many copies of the B cells and high concentration of those antibodies, okay, in the secondary response. But there's something else going on in the secondary response from the memory B cells that's really interesting and a really cool example of natural selection. So these scientists looked at the sequence, the actual sequence of the genes coding for these antibodies from the primary response here and the secondary response. And they had a sequence that they determined by sequencing the gene that codes for these primary antibodies. And they isolated several copies of the antibodies and sequenced several of the genes from the primary response from the B cells that were activated, and all of those gene sequences were identical. They also measured how tightly the antibodies bound to the antigen. In this case, a low number is tighter than a high number. So a low number means it doesn't take much antigen to bind up the antibodies, versus a high number means it takes more and more antigen to bind up the antibodies, so because it's not binding that tightly. So low is good, high is bad. Then what they did is they looked at antibodies made during the secondary response, and they had different sequences, a bunch of different sequences. So this is the same antigen. Now this is stimulated by the memory cells, then we'll make a bunch of B cells after they've been stimulated the second time. But these are not the same sequence as the original sequence. They have some important mutations in there. And again, they measured the affinity for the antigen, and it's pretty variable. So again, big number doesn't bind tightly, small number binds really tightly. And this is a big, big change something like 100-fold difference. Is that right? And yeah, 100-fold difference in how tightly they bind the antibody by these changes in amino acid sequence. So how does that happen? The secondary response involved B cells that had evolved compared to the first response. So if you think of the first set of B cells, you have this population of cells, and they have a certain genotype to them. Turns out all the genotype is the same. And in the second population of cells, you have different genotypes. So the frequency of the alleles, frequency of those genes had changed in the population over time, and that's our definition of evolution. So how does that happen? Okay, so the B cells are, in the initial response, are selected from B cells that already exist, and then if they bind with the antigen, they're stimulated to make by mitosis, to go through mitosis and make a bunch of copies of that particular B cell. So mitosis is a copy machine, and it makes exact replica of the genome of that B cell, and that's why all the antibodies have the same sequence. But in the secondary response that involves these memory B cells, the memory cells had different genotypes, as shown by that last figure. And how do they get those different genotypes? Those different genotypes came from, or resulted in, antibodies that had differences in how well they bound to the antigen. And in the development of the secondary response, a B cell has to bind to an antigen in order for it to get a survival signal, and when it gets that survival signal, it goes through mitosis. So ones that bind really tightly to the antigen in the secondary response get a lot of survival signal, and they get a big signal to go through mitosis. Ones that don't have much affinity for the antigen don't get much or any survival signal, and so they aren't stimulated to go through mitosis. So there's competition for a survival signal. There's genetic variability in the population, and some B cells are selected and others are not selected. So this is natural selection. There's genetically based variability in the population. That variability gives some B cells an advantage over other B cells, and those B cells divide and reproduce more than B cells that don't have an advantage. So where does the variability come from? In your lymph nodes, B cells will travel, these memory B cells will travel, and they'll undergo hypermutation in this spot in your lymph nodes. A set of genes that are specifically cause mutations in B cells are expressed in these lymph nodes, and those genes cause random mutations in those antibody genes, and some of those mutations give the B cell an advantage in that it helps it attract a survival factor and helps it then get stimulated to go through mitosis. Some of those don't make any difference, and some give it a disadvantage in that they're actually worse than the original B cells. And then those cells go through selection because the ones with the highest affinity, by virtue of their mutations, are selected for, and they get to reproduce and they survive. And the organism mounts a more robust response than the first time because they have better affinity now. And some don't, and they die off. So, in relation to the current pandemic, this nice little story is not how it always works. So not all antigens elicit a really good response. I've made tons of antibodies in my lab in my research career, and you're always just guessing. So I make my antibodies in rabbits, and we immunize the rabbits, and sometimes the rabbits are very allergic and make lots of antibody, and sometimes they don't at all. And just like different individuals may be allergic to something or not. So because there's this element of chance and mutation in the production of these antibodies, sometimes you'll have a really big response and sometimes you won't. In the case of COVID-19, most of what is hurting people is not part of this antibody response, but part of your innate immune response. Before you develop antibodies, you have other parts of your immune system that can respond to an infection faster. And those parts of your immune system seem to be, in people who are severely ill, seem to be overreacting. They have too much of a response. And those parts of the system are designed to kill whatever is infecting. And if you turn on the killing machine too strongly, which seems to be happening in severe COVID-19 patients, then the killing machine starts to attack its own tissue instead of just attacking the virus. The stuff that I was talking about here today in terms of what happens with acquired immunity, a lot of times we don't really have the answer on that because we don't have enough information about the kinds of antibodies that people are making as a result of being exposed to COVID-19. The strategy in terms of trying to make humans have antibodies without getting the disease, that's what's going on with vaccines, but we don't have the results of that yet. So stay tuned, and when we get this information next fall about how people are responding and how the immunizations are working, this information will come in handy.